Yes, as Frank said, I'm representing the Northern Gulf of Alaska group, and like the Northeast Shelf, we're one of the 3 new sites. And big site news for us, we actually are having a May cruise right now. There's just 3 of the normal 22 scientists who would be on board. But it's happening, and we hope to be able to get enough data to continue at least the backbone of our 22-year time series of May cruises from this region. And then you probably heard the business meeting. Because of COVID-19, there's our site review and our proposal timing have gotten pushed out a year. Big phenomenon in our region, which is in the Northern Gulf of Alaska, is the long-term warming, and then the recent 2015-16 marine heat wave with a little bit of a reprise in 2019. And there's been, I want to highlight 3 really interesting and somewhat unexpected consequences of this marine heat wave. First, this is a epifluorescence image of this exact same picocyanobacteria that Heidi was just talking about, the little yellow fluorescing dots. These are the smallest primary producers in the ocean. And we're at 60 degrees north, and I never expected to see bloom densities of these organisms in our ecosystem, but that is in fact what we have in the summer, and especially in these warm years. And so that's the first surprise. But on top of that, last summer during our process cruise, we were able to compare primary productivity with export fluxes from sediment trap measurements. And this is Stephanie O'Daly's work, one of our grad students in the program. And you would normally expect a summer small cell dominated system to have very low vertical flux rates. And that's not what we found. We found that the export fluxes were 75% of the primary productivity at a number of our shelf stations. So there's something very non-paradigmatic going on this shelf in the summertime in terms of how the production moving through the food web, which we hope to explore further this summer during a process cruise that we hope will happen. Secondly, we're pulling together information on the planktonic food web from the heat wave, the 2015-16 heat wave. And one of the consequences of that heat wave, as many of you may know, was huge seabird mortalities along the whole Pacific Rim. This is a picture of common murres in Alaska in early 2016. And what it looks like, to make a really long story very short, is that sort of the weak link and perhaps one of the least resilient components of this lower trophic level ecosystem are the euphausiids and the forage fish, which are the small animals that are highly mobile, that are really important prey for seabirds. So while everything changes during the heat wave, this is the component that really seems to almost disappear and be the crucial link to the seabird mortality. And then finally, last summer, we were able to do an extensive investigation of one of the major river plumes in the region. And these rivers are hugely important for supplying buoyancy and the trace nutrient iron to our continental shelf ecosystem. And we found those things, but we also found a number of surprises with respect to this river plume. Higher productivity than we expected, which we partially attribute to shear-induced mixing, and also to decoupling between the phytoplankton prey and some of the grazers that would normally help keep their numbers in check. And we're working with David Hill at Oregon State to improve the representation of this really complex hydrology in our biogeochemical modeling. Okay, so now I'm going to tell you about some of the challenges and how we're meeting challenges. Firstly, it's very large and heterogeneous. So here's an image of surface chlorophyll from Modus Aqua satellite in June of last year. And this is our site basically in this box. And you can see the tremendous heterogeneity from very low to very high chlorophyll in strands and filaments. And I'll point out that the area of our site is about equivalent to the state of Minnesota. And we're trying to cover this in some sense or another in 3 seasons per year. So to help us with this challenge, we are about to implement a glider program. We hope that these gliders will be able to transit the entire area from late winter through late spring of each year. And that's basically ready to hit the water as soon as we can get the people and the instruments safely all into one place and launch it. And then last summer, we were able to use a towed package called an Acrobat to do a really detailed spatial survey of the river Plume. And this is a 3D representation of the salinity that we found last summer. And we're going to be adding some imaging instruments to this towed package to help us incorporate the next trophic levels up, the zooplankton in particular, into our detailed spatial assessment of this feature and others. The weather up there is challenging. This is wave data from a NDBC buoy that's in our study region, and from fall, September through December of last year, with a 2-story house for scale. And I'll point out, this was our fall cruise, so about 1 to 1.5 stories worth of waves. And this is when the transmitter on our mooring got swamped by 30-foot seas in October. So to help deal with this, we've got a couple of strategies. We've added a small boat called the Nanook to our fleet. It can dash out from Seward and take advantage of little weather windows to near-shore sampling. And last summer, we deployed extensive mid-shelf mooring array of 3 moorings that has a whole bunch of sensors, including a wire walker that does vertical profiles twice a day, passive acoustics for marine mammal censusing, active acoustics for zooplankton, sediment traps for export flux, etc. So that's hopefully that's getting serviced right now this week. They're actually hunkered down from a 20-foot wave storm right now, but that was one of the compelling reasons for getting 3 people out there right now, is to get this thing turned around and continuing to collect data. Another challenge is that our model really wants rates and conditions in order to be able to get the right answers for the right reasons. And so we've got some wonderful new investigators in our program that are helping us provide those. And this just shows you who they are, some of their areas of expertise. And then as I mentioned before, we have 3 cruises a year. We are now, as of last year and going forward, thanks to a partnership between the University of Alaska and NSF, we're able to use the RV Sikuliak for 2 of those 3 cruises, which is just huge, because otherwise we're on a small fish and wildlife vessel with basically no lab. And so we're able to do way more process work. And this just shows one of my grad students doing some mixotrophy experiments last summer in the walk-in environmental chamber on that ship. So it's a real boon to our program. And then outreach was a challenge at such a remote site. Last summer, we had our first cohort of REUs come through. We had 5 of them on board for the entire 3-week process cruise. It really livened things up, and it was interesting learning experience for everyone involved. They were great. We have a teacher at sea program. We have a teacher on every cruise. And last spring, Gab's our TC, and she is going to be spearheading our new education and outreach. We're transitioning to a new education and outreach lead from Marilyn Sigmund, who ably led us through the first few years of our program. And a really exciting feature of this is that Katie and the Center for Alaskan Coastal Studies is going to be leading the E&O for both our program and for the Beaufort Lagoons program. So we hope to have sort of a built-in synthesis with that going on. So looking forward, we're excited about starting for real some site comparisons, including through this education and outreach hire that will serve 2 sites, getting more information on some of these unexpected consequences of warming, including with new observation methods and with a better fleet for these process studies, and then really building our existing 2-way integration with the modeling. Thanks a lot.